Thank you for the opportunity to speak here today. First, my disclosures. I'm a co-founder and equity holder in Celsius and Immunitas. And until July 31st, 2020, I was an SAB member in Thermo, Neogene, and Cirrus. From August 1st, 2020, I'm an employee of Genentech. My lab has just moved to Genentech last month, and so the work I'm going to show today is from MIT, the Broad, and HGMI. Now, in many problems in biology, there's an enormous space of possibilities, even though only very few of them actually exist. These include examples like somatic mutations in cancer, regulatory sequences in promoters and enhancers, possible gene expression profiles, and what is my personal favorite, genetic interactions. Now, in each of these cases, the number of hypothetical possibilities exceeds by many orders of magnitude what we could ever do experimentally. But we don't know upfront which options exist and which ones matter. And so how can we study something systematically if we cannot do it exhaustively? Well, historically, what we did as biologists was to use informed design in order to limit the experimental search space with things like selected probes and design promoter sequences and gene signatures and so on. And these are all great approaches, but they don't fully solve our problems. We're not sure at the end that we know the general answer by looking in these limited ways. So can our experimental designs be revised to either be inspired by algorithms or used in ways that we can learn more from less? As an intuitive idea of how I think about this, I'm going to use a little example from art. So imagine that functional discovery in biology is about figuring out which painting I'm hiding in this frame. Now, every experiment means uncovering one tile, and I can only uncover some tiles, far fewer than the full number of tiles. Now, I know something about this picture, though. It has patches of color, which is what I want to know, and lots of uninteresting background. Until recently, all we could do was remove a few tiles, so we had to focus. If we already knew that there was some color, for example, here in red, then the wise thing to do was to look around it, which let me uncover a red patch. Or here I had yellow, and I could dig around for some yellow patch. If instead I used the exact same number of tiles and threw them randomly at the picture, I wouldn't have figured out much at all. I found one blue tile and nothing else that was blue. But what if I were allowed a lot more tiles? Now, even though I just threw them randomly at my image, I can actually tell that this is a Miro painting. Now, this makes perfect sense in machine learning. We do this all the time. But doing this in the actual biological experiments is something that's not been as typical, although it has been done before. And so a good recent example of this has been the development of massively parallel single-cell RNA-seq in methods like DropSeq. Early on in single-cell RNA-seq, we learned that the method samples only a small portion of the RNA in the cell. And we were faced with a design for inference problem. Should we invest our resources, which means experimental effort and sequencing reads, at getting more information per cell, or should we get more cells? Now, methods like DropSeq favor sparse, noisy data from a massive number of cells over richer, more precise data from a small number of cells. Now, because expression within cells and patterns across cells are highly structured, it turns out the algorithms can leverage this structure in order to infer meaningful biological representations of each facet of a cell's identity. Now, over the last several years, we started developing additional kinds of designs for inference. And what I'm going to do in my talk today is give you several such examples. And I'm going to focus on the circuits that control gene expression and act through regulatory sequences that affect RNA production. Experimentally, we can use genetics in order to do perturbations by intervening in one or more component at a time, and then measuring the effect, for example, on gene expression. As my first example, I'm going to focus on cis regulatory sequences. And this was work that was started and led by Carl de Boer, who was a computational postdoc in the lab and now has his own lab in the University of British Columbia. One way to learn how regulatory sequences drive expression levels is to train a model that's based on looking at many pairs of examples of a regulatory sequence and the associated expression. To get these examples, people typically do one of two types of experiments. Either they use native sequences. We know that these sequences are functional, but they tend to be limited to examples in nature. Or we can design sequences and test how they drive expression. But synthesis technology currently limits the numbers of examples that we can look at. And so Carl asked, could we generate much bigger data if we just used random DNA? Why could this possibly work? Well, transcription factors bind very short and partly degenerate sequences, and so most transcription factor binding sites would be very prevalent in random DNA. In general, a motif with X bits is expected to occur at 2 to the X minus 1 bases. And so in a library of, say, 10 million 80 base pair long random sequences, we will have at least 40,000 examples of each of 90% of yeast transcription factor motifs. And for 50% of them, we should have millions of examples for each of them. Now, because random sequences are extremely easy to synthesize, we should be able to produce lots of training data. But of course, this has to actually work. And so to test this idea, Carl devised a simple assay, which he called the Gigantically Parallel Reporter Assay, or GPRA. He made a library of hundreds of millions of random sequences, each of them 80 bases long, driving YFP from a core promoter construct, put it into yeast, grew the yeast, sorted them by expression, and sequenced from the beans. But there are two potential issues. First, because there are so many N to the 80 sequences, we expect each sequence to appear at most once in the output. This means that the data will be extremely noisy and sparse. Second issue is, who knows if the random sequences will generate any expression at all? So to look at this, Carl grew the library and sorted the yeast into 18 expression beans. This will let him see if there's a range of expression. Then he recovered the live yeast from each bin, regrew them, and measured their expression again. This would let him ask if the expression is reproducible. And here are the results. These are the regrown yeast, but they're colored by the expression in the original bin from which they were grown. And you can see the gray distribution also, which is the natural yeast promoters. And what you see is that we got a broad range of expression levels, as broad or broader than the natural promoters, but also it was reproducible. The regrown yeast expressed at the level of the bin from which they were grown. So now we were ready to do an experiment. We used the assay to measure about 100 million promoters in 2 core promoter contexts and in 3 carbon sources, glucose, galactose, and glycerol. And then we took the data and we first learned an interpretable model of gene regulation, which is explicitly defined in biochemical terms. This model takes a sequence, identifies binding sites for a transcription factor, sums up the sites to estimate the binding of a transcription factor to the DNA overall, predicts accessibility for a transcription factor to bind chromatin, and finally predicts the expression level. And I want to highlight a few features. We have motifs that can be either initialized with known motifs or learned de novo. We have transcription factor binding, which is summed up by what is called the billboard model. And also a transcription factor's ability to actually bind and activate expression is also impacted by accessibility to bind chromatin. And then finally, this can be potentiated by binding of other transcription factors. Putting in all these biological details ends up in a model with between 1,000 to over 200,000 parameters, depending on whether we initialize the motifs or learn them de novo. And we can fit this because we still have far more data than that. Now we trained the model on our data and we asked, how does it do at prediction? For this, we first made an independent test set of another 10,000 random sequences, but each of them is measured in 50 cells per sequence, whereas before we would have usually 1 cell or less per sequence. So we get high quality data. And the model predicted these at 92% accuracy. We also took 80 base fragments of native yeast promoters. And again, the model predicted well, now at more than 85% accuracy. So now we kind of had basic faith that this model can generalize. And we asked about biological lessons, and I'll highlight one. We decided to turn to a specific problem that is related to the position and the strand preferences of motifs. So in past experiments, like this pioneering work that was done in Eran Segal's lab years ago, people tried to learn them by collecting designed experiments. They tiled each transcription factor binding site across one or a few background sequences in the MPRA, basically moving the site one or a few bases at a time on a fixed background, measuring expression each time. But as you can see, most of the time, the expression levels are really erratic. Even a tiny move by 1 or 2 bases changes expression a lot. And the model didn't explain this well. Now, position and orientation should be well covered in random DNA. So to see if our model can help we devised an extended model that fits parameters based on location and strand of the motif. And this brings us to about 220,000 parameters when we initialize with known motifs. And we repeated now these types of tiling experiments. We slid a motif, here for MGA1, on a random sequence background. And just like in the previous experiments from the Sega lab, we see that the measured expression, which is in black, was jagged or erratic. Even 1 to 2 base shift had a big impact. But our model in red predicted this extremely well. And we saw this in experiments tiling each of 6 motifs in 3 random contexts. Now, because our model is interpretable, we can ask which features relate to this behavior. The positional activity of MGA1 mostly doesn't actually correlate with this, but the learned accessibility does. So what is going on? It turns out that these changes in accessibility or potentiation and expression are actually coming from multiple weak sites for other transcription factors that get created or disrupted as we slide the primary MGA1 sequence. None of these sites is considered by classical models. They are too weak. They are too weak to be learned from the small data sets, but they are not too weak to matter for biology. So with this in mind, we asked if we can understand what is the contribution of strong versus weak sites on each gene's expression in yeast. For this, we ran a computational experiment. We computationally deleted each transcription factor, and we asked what is the impact on the expression from each native yeast promoter sequence, and we called this the interaction strength. And it turns out that only 0.1% of possible regulatory interactions are predicted to alter expression by more than 2-fold. These rare strong regulatory interactions do explain a disproportionate amount of expression, but at the end of the day, 94% of expression is attributed to the much more prevalent weak, meaning less than 2-fold change of regulatory interactions. And so to summarize so far, I showed you that because functional transcription factor binding sites occur frequently by chance, we can devise an experiment, the GPRA, to measure the expression levels associated with hundreds of millions of random sequences. Random DNA gives a broad range of reproducible expression, and we can fit great models from it. A biochemically interpretable billboard model explained up to 92% of the variation in test data of random sequences and 85% of native sequences. I didn't show you, but it also learned motifs de novo. It correctly predicted chromatin structure, and it identified transcription factor regulators of chromatin state. Then we added positioning to the model, and we showed that this model predicted the impact of tiling motifs and showed that the erratic behavior is because of the creation and the destruction of multiple weak interactions. Then when we analyze native sequences, we see that these kinds of weak interactions are numerous in every promoter, and they're prevalent across promoters. And this has evolutionary implications, some of which Amos Tanai actually highlighted in seminal work in 2006. Now, we're working in parallel on adaptation for human cells, but for now, I'm going to stick to the theme of evolution. And so this work suggested to us that maybe we can take on other problems in gene regulation, where we need to capture a large sequence space. And so the second part of the work was led by Ishit Vaishnav, who's a grad student in the lab, together with Carl. And they focused on gene regulation in the context of evolution. So in trying to understand the relationship between sequence and fitness, people often replace direct measures of fitness, like growth rate, with molecular proxies, like gene expression levels, basically breaking the problem in two. Have one function from sequence to molecular proxy, while assuming that there is a second function from the proxy to organismal fitness. But usually we can sample just bits and pieces of the vast sequence space, starting close to no sequences, and usually starting known to close sequences. And so that reduces our ability to build comprehensive models. So we thought, can we use the ability to learn from GPRA data, these sequence to expression models that generalize really well? Maybe they generalize to any arbitrary sequence. And in this way, we can construct the sequence to expression function. And then we could use this model in order to ask relevant evolutionary questions. But in this case, because here our goal is to have a model that generalizes as well as possible, And so we turn to deep learning models, especially to deep transformer neural networks that tend to perform really well for prediction tasks. We were willing to let go of the interpretability, but we were still inspired in our modeling by biological ideas. We tried to frame models that maybe would be able to capture things like motif specificity and interactions and transcription factor scanning and long-range interactions and activity post-binding and so on, but we can't really tell at the end in any explicit way if those are captured. This model has 3.3 million trainable parameters, but our data is still far bigger. And so the transformer-based model had excellent predictive performance, 98% accuracy on the new test data set of random sequences and 96% for native yeast sequences. And so this means a reduction of 33% and more in the error compared to our best performing biochemical model. Now, one immediate utility is that we can use this model just for promoter design. We used the trained model to search for sequences that it predicted would have especially high or low expression. And then we synthesized them and measured experimentally. The blue distribution is the measured expression of promoters designed to have low expression, and the red is the same for high. And as you can see, they had the desired expression, even going beyond the native extremes. So next, we took our highly predictive model to tackle evolutionary questions. The first one was whether we could transverse evolutionary trajectories under different regimes established in the literature, like random drift or strong selection with mutation. For random drift, we take a random sequence, introduce a random mutation, calculate the predicted change in expression by the model, and then repeat the process, doing n mutational steps to generate one trajectory. And you can see that if you look at the expressions change in each step for each of 5,000 trajectories, the model predicts a rapid increase in expression divergence, so that by 32 mutations, we get nearly as different expression from the original sequence as from an unrelated sequence. And we validated the trajectories experimentally by measuring the expression of the sequences in each mutational step in our assay. Now we look at strong selection weak mutation, or SSWM regime. And here the mutation rates are low enough to assume only one mutation per sequence per step, and the selection is strong enough for mutations to fix in the asexual population. So we start with a set of all native sequences, and at each step, for given starting sequence of length L, we consider all of its 3L single base mutation neighbors. Then we use the model to predict their expression, and we take the sequence with the largest increase or decrease in expression as the starting sequence for the next step. We repeat this for 10 to 11 steps, and the simulations show that we reach extreme expression within 3 to 4 mutations, regardless of the starting point. And experiments with the same sequences validate this. Now we can ask what would happen if the promoter needs to satisfy not just one, but maybe two opposing expression requirements, say high expression in one growth condition, low expression in another. This is what happens in reality with a gene like URA3 in yeast, where high expression gives you high fitness in defined media without uracil, but it's detrimental in complex media with 5-FOA. So we simulated this with the same setup, except that at each step, we calculate expression using two different models that were trained with data from two different media. And then we chose the sequence that maximized the difference in expression between these two environments. And with these conflicting objectives, expression changes are slower and they require more mutations. And one interesting thing in this scenario is that by 10 generations compared to the starting sequences, the evolved sequences became enriched for motifs for transcription factors that are involved in nutrient sensing and metabolism. Then for the last regime, we looked at stabilizing selection, which favors maintaining the existing expression levels. And we asked what happens to regulatory mechanisms. So we initiated with sequences that, based on our original biochemical interpretable models, will have high or low regulatory complexity, which is 1 minus the Gini index of the TF interaction strength. Then, like before, we're going to test each random mutation, and we're going to select from them the sequence that maintains the expression level. And we validate this experimentally. And now what we find is that even though expression levels didn't change, the regulatory complexity shifted from high or low complexity as the starting point to an intermediate complexity in the endpoint. So by 32 generations, it's much closer to the average complexity that we see for both random and native yeast sequences, which are in the grays. So this may mean that the regulatory complexity we see in native sequences could just reflect the space of possible configurations with equivalent expression outcomes. Okay. Now let's move from these idealized regimes to see if our model can help us understand the imprint of selection in naturally varying sequences. For this, we used the great data from the Peter et al. Paper, which sequenced the genomes of 1,011 Saccharomyces cerevisiae strains. And we wanted to use our model to build a score of signatures of selection on regulatory sequences. As an analogy for what we did, think about how we use the genetic code as the basis for a score like DNDS for selection on protein sequences. Now, how do we do this? First, we're going to take a collection of orthologous native promoters from the strains, and we're going to use our model to predict expression for each of them. This is going to give us one variance that we will call sigma B. Next, we generate another set of sequences by adding random mutations to the natural sequences while keeping an overall matched relative Hamming distance an overall matched distribution of relative Hamming distances. We then are going to use the model to predict expression for these generated sequences, and this is going to end up with sigma C. And then finally, we calculate the expression conservation coefficient, or the ECC, as the log odds of sigma C and sigma B. And this means that positive ECC values, where we have a higher variance in the generated than the real sequences, would suggest stabilizing selection. And they're indeed enriched for genes involved in conserved cellular processes like RNA and protein metabolism. And then we would have negative ECC values. That means higher variance in the real than the generated sequences. And that would be a signal of diversifying or positive selection. And indeed, we see those negative ECC values enriched in genes involved in metabolic processes that vary across the yeast. And just as an example of a predicted positive selection, here is the CDC36 promoter. There are 19 alleles amongst the 1,011 strains. 2 of these alleles are predicted as low expressing and independently evolved, and each has a different mutation in one of the 2 critical bases of the UPC2 motif. Okay, now to see if the ECC is consistent with the divergence that we observe in actual expression levels, we also used cross-species RNA-seq at different evolutionary distances. We had data sets within Saccharomyces strains, within Ascomycota yeasts, and within mammals. And in each case, we identify orthologs for Saccharomyces cerevisiae genes, calculate the ECC based on the promoters of the 1,011 Saccharomyces cerevisiae strains. And on the other hand, we're going to use the cross-species RNA-seq of these orthologs in order to identify which genes have significantly conserved expression, either within Saccharomyces or within Ascomycota, or even within mammals, and which have significantly divergent expression. And then in each case, we find the genes with a conserved expression have higher ECCs than those with divergent expression, going all the way to mammals. We also compared the ECC values with the effect of the change in expression on actual organismal fitness. For this, we looked at 80 genes where Eran Segal's lab in another study measured their fitness, which is on the y-axis, at different expression levels of the gene on the x-axis. And that I'm showing this here for 4 of the 80 examples. We needed a statistic of fitness responsiveness, and for this, that is for how much the fitness is affected by changes in expression. So for this statistic, we calculate something called the total variation for each of these expression fitness curves. This statistic goes from 0, when there is no relation between expression and fitness, like ADR1 at the bottom, all the way to 1 when the dependency is high, like RAP1 at the top. And the ECC is highly correlated with this fitness responsiveness, meaning the genes where changes in expression impact fitness in this lab assay also have higher ECC by our calculations. So a gene's ECC captured some imprint of the history of the regulatory sequence. Now we wanted to complement this with how future mutations may affect the gene's expression. So we looked at its mutational robustness. We defined mutational robustness for a sequence of length L as the percent of the 3L single nucleotide mutational neighbors that are predicted by our model to have a negligible expression change. And the mutational robustness was correlated with both fitness responsiveness and with the ECC of the 80 genes tested by the Sega lab. And in addition, genes with conserved expression across Saccharomyces, Ascomycota, or mammals had higher mutational robustness. So finally, we asked if we can find an effective way to characterize the full landscape and visualize it in low dimensions. For this, we're going to encode for each sequence its evolvability, which we define as the extent to which mutations in the sequence would change its expression phenotype. So we take a given sequence of length L, and for each position, consider each of the 3 possible mutations, and we calculated the predicted change in expression using our model. Then we get a vector of length 3L, where each entity is the change of expression. We take this vector and we sort it, and this gives us the evolvability vector. And now we take the vectors for n random sequences, and we use an autoencoder, this is evolvability vector, and we use an autoencoder in order to learn an embedding of the sequence to a latent representation in 2 dimensions. But we do it in a way that constrains it to a limited number of archetypes using a nice method developed by Smita Ramaswamy's lab. In this archetypal latent space, the evolvability vector of each sequence is a single point within a convex polyhedron whose vertices represent the evolvability archetypes. These are extreme canonical patterns, in this case of expression change in mutational neighborhoods. The best fit in our case was to 3 archetypes. And what I'm showing you here are the native yeast promoter sequences. These are the black dots projected into the archetypal space that we learned from random sequences. And if we looked at the evolvability vector of the yeast sequence closest to each archetype, we see that 2 archetypes correspond to local expression minimum or maximum. These are sequences where most 3L mutational neighbors do not change expression, and the ones that do either increase it in A minima or decrease it in A maxima. And then the 3rd archetype is plastic. Most of the 3L mutational neighbors change expression and are equally likely to increase or decrease it. And in addition, there's a 4th interesting location, which is this cleft, where we have a concentration of those sequences like S4 that are mutationally robust. And this cleft is enriched in native sequences. So the archetypal space captures many features that I've shown you by now for native sequences, including their expression and their ECC. And finally, we embedded all the extant variants of each yeast promoter sequence from the 1011 strains into the same space. And when we examine a sequence like the DBP7 promoter from the S288C strain, we see that it is close to the plastic archetype. The other 14 natural alleles are highly dispersed across the space, but not in the robust cleft. And when we start with a promoter like the one for UTH1 that is close to the robust cleft, so are the 21 alleles for it, which are all one on top of the other. And so overall, the extent of dispersion really matches the archetypes. And so to summarize this second part, I showed you how we use GPRA training data with random sequences to learn a deep transformer neural network model of sequence to expression. This model generalizes extremely well. It lets us engineer promoter sequences. We use the model to look at questions in evolutionary biology. First, we simulated and experimentally validated how expression changes under different kinds of regimes. And then we developed the ECC as a statistic for detecting selective constraint on gene expression from naturally varying sequences, something analogous to the NDS. And then we showed that the ECC agreed with expression diversity in cross-species RNA-seq across 3 different clades, and with measured expression to fitness curves. And then prospectively, we can use the model to calculate mutational robustness. And we showed that the current regulatory sequences are often also mutationally robust. And then finally, we developed a representation of regulatory sequence evolvability, highlighting these 3 evolvability archetypes, as well as the robustness cleft. And we think that this approach should be applicable in other systems and organisms. Okay, so far we looked at cis regulatory sequences. Now, let me turn to programs of co-regulated genes. And the second part is all work by Brian Cleary, who was a graduate student in the lab, but is now an independent Broad Fellow with his own group. And what Brian reasoned was that maybe we should not need to measure actually each gene individually, especially in cases when that's difficult for us, and we could still recover the information that we need. So for example, what if instead of measuring each gene separately in each sample, we could collect compressed data on gene expression and then use an algorithm to decompress it and give us all of the genes later. This is based on advances in the area of compressed sensing over the last 15 years or so. And that's worked well for things in other fields like images because they're structured and they're not random. And so gene expression is also very structured. Sets of genes are often co-regulated, going up and down together in sets of samples. And these modules are often used in different combinations, like pieces of Legos, to give us the cellular state. And so could we somehow compress gene expression measurements too? So if we could do this, it would actually be really helpful because there's many measurement methods that have limited measurement channels. They can measure the abundance of, say, only dozens of RNAs and proteins. But what if we could use the same 100 measurement channels that the instrument allows us to study the level of 10,000 RNAs or proteins? How would we do this? Instead of measuring individual genes, we imagined measuring the abundance of a much smaller number of composite genes. The composite gene is a linear combination of different gene abundances. And the coefficients can be chosen randomly. And important, you can show that they can be chosen binary, which is easier to implement in an actual wet lab. So imagine for now that we can collect this kind of compressed data. How will we decompress it so that we know the level of each gene? One way to do it is using a little bit of training. First, we'll use, say, 5% of full profiling data to fit a model where the expression of genes across the samples is explained by a dictionary of gene modules and by the activity levels of modules in the samples. And then we can use known factorization algorithms for this, like SVD or sparse NMF, but we actually also developed our own algorithm, sparse module activity factorization, or SMAF, which is sparse in both the module dictionary and the module activities. Then we can take the remaining 95% of the samples, assuming they are measured only in compressed form. And in each case, for now, we're just simulating compressed acquisition with random weights. And then we will use the module dictionary that we learned in the first step from full profiles and the compressed measurements in order to predict the levels of all of the genes or to decompress the data. And in a test like this with simulations, we can then compare our decompressions to the original data and see how well we did. And we saw using these kinds of simulations of compressed measurements on both bulk and single cell RNA-seq data, that compressed measurements, first of all, in still compressed form, correctly cluster profiles, even when we introduce noise, and they can be decompressed to get pretty correct abundances. Now we can ask if we can even decompress the compressed measurements without any training. That is, even if we have never seen a full profile. This is called blind compressed sensing, or BCS in the literature, and it requires us to make some assumptions. The assumptions that were made in other fields are not good for biology. So instead, we assumed that samples that group together based on compressed measurements are likely to use similar active modules, even if we don't know what the modules are. And so Brian devised another algorithm he called BCS SMAF that first clusters the compressed samples and then searches for small dictionaries of modules per cluster, and then concatenates them as a starting point. And from here, we basically use iterative optimization on the modules and their activities. And so could this possibly work? Here is the original full profile data, 14,000 genes per sample. BCS math saw only 20x or 50x compressed composite measurements, that is 700 or 280 measurements per sample. And it still recovered profiles with up to 70% correlation with the true abundances. But this is all theory, it's all simulation. It's not theory, it's all simulation. Would it actually work in practice if we came up with the right kind of lab method? Can we do this in the lab? So we focused on assays that can measure only a small number of individual RNAs. And we came up with a two-step design where we use single-cell RNA-seq data to find gene modules and define compositions. And then we perform composite images of these combinations, recover the module activities, and decompress the image to the level of individual genes. To do compressed measurements, we measure in each color here, in each cycle, multiple transcripts together, simply by using probes with a single color targeting multiple genes. And together with Fei Chen, we measured 9 composite images of 37 genes in the mouse cortex. And as you can see, there is very little pattern that's apparent to the eye using these composite measurements. Next, we decided to decompress these images. And we used 2 approaches. In one approach, we first segment the cells, and then we decompress the expression per cell. But we also developed a segmentation-free decompression, which is based on an autoencoder approach. First, we do the decompression from 10 to 37 channels in the encoded latent space, and then we decode the images. And when we decompress, we get very precise and accurate patterns, which I'm showing you here for 3 genes. The images you're seeing are not directly measured. They're decompressed images. But the pattern fits well when we compare it to the Allen Brain Atlas on the left. And also when we actually individually measure genes on the same section at the end of the process. The red signal is the directly measured level of this gene, just with 1 probe. The green is recovered from decompression, and the yellow pixels, which is most of what you see, are the overlap at the pixel level. So finally, we extended this to a larger data set. Again, we got good reconstruction after decompression for both cell type and cell state genes. And we also recovered cell cluster labels and spatial patterns. And we gain here 100 to 300-fold in efficiency, because we don't only just multiplex genes, but we also no longer need to do spot detection. And so we can use just 20x imaging. Okay, and so to conclude this 3rd part, I showed you that because gene expression is highly structured, we should be able to use compressed sensing by measuring random composite measurements. This required us to have shared regulation between genes. And there are cases like cis-EQTL, when a gene is very specially and separately regulated, so this method will not work. But for much of gene regulation, it is shared in these nice modular structures. And because of this, we can decompress from 200-fold compression using a 5% training set, and even apply decompression to compressed measurements blindly with no training at all. And we also applied this experimentally, showing compressed imaging for RNA. And we have a segmentation-free decompression method for this. And in addition to the 4-fold more multiplexing and growing, we can work at much lower spatial resolution. And overall, this gives us between 100 to 300 gain overall, and that gain is grown. So finally, in the last part of the talk, I want to turn to the problem of the regulatory modules of genes that control the expression of other genes and the genetic interactions between these regulators. And this part is still work in progress. It started with work done by Trey Dixit when he was a computational grad student in the lab, and Oren Parnas when he was an experimental postdoc in the lab, and now has his own group at the Hebrew University, and more recently by Brian Cleary in one line of work, as well as Katie Geiger and Bashak Erslan, who are postdocs in the lab in another line of work. And so first, a quick reminder of how we assess the regulatory function of one gene at a time. Say we wanted to find all the genes that individually can affect the expression of gene X. For this, we use the pool genetic screen. We deliver a set of barcoded perturbations, usually guide RNAs with CRISPR, such that each cell would receive at most 1. Then we grow them, and then at the end point, we sort the cells by the expression of the gene X we're interested in. We sequence the barcode, and we look for enrichments. And this design, the pool genetic screen, is one of these amazing experimental designs, but it does have 2 issues. First is that we had a very simple readout from each cell, the level of expression of one gene X. And so we have to choose X ahead of the experiment. And at the end of the experiment, all the hits will appear the same. They affected gene X. Second, what if we assay the effect of more than one perturbation at once? It may be that these genetically interact with respect to their effect of some genes, here at yellow, but not on others. So choosing one gene could give us one answer, and choosing another gene will give us another answer. To get a full answer, we need a full profile. And so a few years ago, we began to tackle this by devising a lab method we called Perturb-seq, which combines a pooled CRISPR screen with single-cell RNA-seq as the readout. We deliver perturbations to cell. We make sure that they get expressed so that they are barcoded basically as RNAs. And after the experiment, we profile by single-cell RNA-seq and recover which cell had which perturbation and which profile. And one of our first applications was for immune cells that we study called dendritic cells stimulated with LPS. And we used a regularized linear model to recover what we call a regulation matrix of the regulatory coefficients of each guide in the column across each gene in the rows. Blue is a negative effect, red is a positive effect, and the guides and the genes are clustered. The genes partition into programs that are affected in similar ways by the perturbations. And that fits nicely into the biology of dendritic cells. And the perturbed transcription factors here grouped into modules of factors that, when we perturb them, lead to similar impacts, again, in ways that largely make sense. Perturb-seq turned out to be pretty scalable. A power analysis of these first screens, which were saturated for cells and sequencing depth, showed that for a state or a signature level effect, it's enough to have as few as 10 to 30 cells per perturbation and a few hundred reads per cell. And as a result, it's now used to understand all sorts of effects in vitro and in vivo, encoding and non-coding variants, one cell type, multiple cell types, and so on. And so that addressed the first question of having a high content readout and not having to choose one reporter, and not all hits are the same. But does it really address the genetic interaction problem? And so, for example, based on single perturbations in the dendritic cells, there's an inflammatory program which is activated by REL-A, one transcription factor, and repressed by NF-kappa B1, another transcription factor. But what would happen if we perturb both of them at the same time? Well, naively, we can use Perturb-seq for this to assess the impact of combinations of perturbations, simply by going to a higher multiplicity of infection, so that some cells will receive 2 or more perturbations, and learning a model with interaction terms from the cells with multiple perturbations. Not a big deal to add that, and you do get answers. We will be able to determine the impact of each pair of perturbations on each gene, and you can categorize them in this way by the extent of interactions that they have in each of the 27 possible categories for 2 interacting genes. And when we do this, this showed us that just for this pair of transcription factors, the extent and even the type of interaction depends on the target gene. So even in just the inflammatory program, and just for those genes where NF-kappa B1 is activating and REL-A is repressing, for some genes there is an interaction in which NF-kappa B1 is dominating, but for other genes, there is no genetic interaction, okay? There is no genetic interaction. You see that the overall effect is just the sum. And we obtained very similar results for several other pairs of transcription factors. And as a result, for each pair of transcription factors, we can quantify their relative impact in each of these class of interactions. So technically, we can do such experiments, but we would still not be able to do a full combinatorial screen of, say, all the 2, 3, 4, and 5-way combinations, even across just 1,000 genes, let alone the whole genome. That one experiment would require 10 to the 15 cells. So is there some way of shrinking this problem and still making some progress and getting our answer? And this brings us back to this regulation matrix and how structured it is in both the perturbed genes and the responding genes. So could we use this structure to make more efficient experiments by using similar insights to the ones we had before, either no longer measuring, say, individual genes and individual perturbations? Maybe we could measure their compositions, or by filling in for experiments that we never performed using the structure that we identify using a limited set of measurements. So for the very closing of this talk, I'm going to show you a brief glimpse of ongoing work with both of these approaches. First, focusing on the first concept, we used droplets to overload the cells such that we profile the sum of their expressions. As a test experiment, we perturbed each of 600 genes in an inflammatory response, either in the traditional way, one cell at a time, or in this compressed way. And we used compressed factorization to learn which modules of co-functional perturbations regulate which modules of co-regulated genes. We're just lucky that somebody came up with a great algorithm for that and we used it. And then the first sanity check that we made is to see that the decompressed expression is 97% correlated between the traditional and the compressed experiments. And then when we look at the impact of 5 major positive and negative regulators in the pathway on 4 major known targets, those are also consistent. And very similarly for the perturbed genes with the top 15 effects. And even if we go further down our ranking to genes with weaker effects, although we see that the signal is slightly degrading, we still see highly similar patterns and effects. It actually works. Another possibility is to use the traditional perturb-seq measurements, but try to predict experiments that we did not perform. So here is a screen that's focused on E3 ligases in immune cells, the same dendritic cells you saw before. We profiled more than 1 million cells, where 500,000 are single knockouts over more than 3,000 perturbations targeting 1,000 genes, with an average of 80 cells per guide. And the rest are double knockouts or more, and they have 0 to very few cells per combination. Just like before, the single perturbations form structure, very clear modules shown here as an embedding based on single cell profiles of cells perturbed by 1 perturbation at a time. But the question is there something that we can do for the combinations? So what we realized here is that instead of predicting the impact of combinations of pairs of genes, we use the structure in order to learn a model that categorizes and predicts the impact of genes from pairs of modules, either the same or different ones. And we found that the fold change we predict in this way correlates well with the observations on held-out data. And so to conclude this final part of the talk, I showed you perturb-seq, a method for pooled screens with single-cell RNA-seq readout. It allows us to screen single gene functions and combinations of them. And most importantly for us, the responding genes form these co-regulated programs, and the perturbed genes form these co-functional modules. And this coupled structure can be leveraged in multiple ways in order to help us take on genetic interactions. I showed you 2 different aspects that we're pursuing. I showed you a quick glimpse into composite perturbations, which reduces the costs and increases the scale of the experiment, as well as and I also showed you using multiple perturbations per cell and the structure from those in order to infer unobserved interactions. And there's more along those lines in both of these approaches. And we also believe that similar approaches are now becoming possible in human genetics. And so with this, I will conclude. I showed you some examples of our work to develop new experimental designs for inference and how the sparsity and the bigness and the structure of biological systems can be used to take on some of the less tractable problems. And I really want to conclude by thanking the people who did the work. The GPRA work was initiated, pioneered, and led by Carl de Boer when he was a postdoc in the lab, and he now has his own lab in the University of British Columbia. And then the work on using these kinds of models, including transformer models, and applying them to evolutionary questions was led by Ishit Vaishnav together with Carl. Ishit is a grad student in the lab. The work on composite transcriptomics, on compressed sensing in general and composite transcriptomics in particular, was led by Brian Cleary, with the experimental work done in collaboration with Fei Chen's lab. And that also extends to the work on composite perturbations I showed at the end, also initiated and led by Brian Cleary, who started as a grad student in the lab and is now an independent Broad Fellow. Perturb-seq was led by Atrey Dixit when he was a grad student in the lab, and Oren Parnas when he was a postdoc in the lab. Atrey now leads Color Genomics, a tech startup, and Oren Parnas has his own lab in the Hebrew University. And finally, the work on the E3 ligase combinations is being led by Katie Geiger, a postdoc in the lab, Shaker Arslan, a postdoc in the lab, as well as Karen Young, a joint grad student together with Caroline Uller's lab. I thank all of them, and I thank you for your attention, and I'll be happy to take any questions. Thank you very much.